Welcome to the Deep Dive. This is where we tackle complex topics and hopefully make them really click for you, drawing on insights from key source materials. Today, we're continuing our journey within the genomic medicine program. We're digging deeper into how our bodies actually function, beyond just the basic DNA sequence. Our focus, a really dynamic, fascinating area, histone modification.
That's right. And understanding this level of detail, these molecular mechanisms, it's really not just academic. It's becoming fundamental if you want to grasp where genomic medicine and personalized health are actually going. These modifications, they're essentially a sophisticated control layer dictating how our genetic blueprint is used. It shapes pretty much everything.
Like cell identity.
Cell identity, yes, how our bodies respond to the environment, even our risk for certain diseases. It's all interconnected.
So our mission today in this deep dive is to really unpack Chapter 7, the one titled Histone Modifications, from our main source text on molecular medicine. We'll weave in other relevant bits too. We want to demystify how these, tiny chemical tags attached to the proteins that package our DNA, how they act like this dynamic control panel for gene expression.
Exactly. And by the end of this conversation, you should have a much clearer picture of how simply wrapping DNA around proteins and then subtly tweaking those proteins influences, almost every aspect of our biology. It's how a kidney cell knows it's a kidney cell, not a neuron. It's also tied into how our bodies seem to remember things and how diseases, like cancer, can actually get started.
And the aim here, like always, is to give you that shortcut so you don't have to wade through dense textbooks. You'll walk away with a solid, informed view on a really critical part of how your body works, way beyond just the A's, T's, C's, and G's of your genetic code. Okay, let's dive in. All right, let's start with maybe one of the most basic but still kind of mind-blowing questions in biology. If pretty much every single cell in my body, in your body, has the exact same set of genes, around 20,000 protein-coding genes, how can a kidney cell be so incredibly different from, say, a skin cell or an immune cell? They've got the same DNA blueprint, right?
It's a brilliant question, and it really gets right to the heart of epigenetics. The answer lies in this concept of the epigenetic landscape. So think of your DNA, your genome, as that master blueprint for building a house. It's got everything specified. But the epigenome, that's more like the architect's annotations or maybe the foreman's instructions on site. It tells the builders which parts of the blueprint to use, when to use them, and maybe how intensely. It's about modifications on or around the DNA that control gene activity, crucially, without changing the actual DNA sequence itself. It's like the operating system for the cell, deciding which programs run.
Okay, so it's not changing the letters in the book, but adding sticky notes or highlighting certain passages.
That's a great analogy, yeah. And you see this incredibly vividly during embryogenesis, during development. You start with a single fertilized egg, an embryonal stem cell, that technically has the potential to become any cell type, pluripotent we call it. But then really quickly, there's this amazing, highly orchestrated process of differentiation. Cells start specializing. This involves massive epigenetic programming, setting up specific chromatin structures. It essentially directs these cells down particular paths, committing them. You become a muscle cell, you become a neuron, you become a liver cell.
And once they're committed, it's generally a one-way street, right?
Generally, yes. It's like that train switching tracks analogy. Once it's on the track for kidney cell, it tends to stay on that track.
And this differentiation, this specialization, is not just something that happens once when we're embryos. It keeps happening.
Oh, absolutely. Constantly. Think about tissues in your adult body that have high turnover. Your bone marrow is turning out blood cells. Your skin is constantly renewing itself, the lining of your gut. These tissues have resident stem cells, adult stem cells, that are always dividing. And when they divide, some daughter cells differentiate to replace the specialized cells that have been lost or worn out. Your immune system is a prime example, constantly being replenished. All of this relies heavily on that epigenetic programming being maintained and executed correctly. It's how tissues keep their identity and function over your lifetime.
Okay, so if I'm following this, while we each have just one genome, one DNA sequence, we actually possess hundreds of different epigenomes.
Precisely. Think about it. We have, maybe around 400 distinct tissues and cell types in our bodies. Each one has its own unique epigenetic signature. Each cell type is using a different subset of those 20,000 genes. What stops your liver cell from suddenly trying to fire neurons? It's the epigenome. It locks in that cell's identity, ensuring it performs its specific job. It's the key to maintaining order and complexity in a multicellular organism like us.
That really clarifies things. It's the software running on the hardware, in a way. So stepping back from cell identity, what does all this mean for us, for our health, for disease risk?
Right, the bigger picture. If you zoom out, epigenetics, along with perhaps the immune system, really forms a foundational pillar for understanding so much of human biology. Everything from development, embryogenesis, through maintaining balance, homeostasis, day to day, and even into the processes of aging. And here's a crucial point. Most of the common non-communicable human diseases, think cancer, metabolic syndrome, heart disease, many immune disorders, they aren't just genetic. They almost always have both an inherited genetic component, a predisposition perhaps, and a really significant epigenetic component. And that epigenetic part often interacts strongly with our environment, our lifestyle.
So it's nature and nurture, but nurture is actually writing notes on the nature, so to speak.
That's a very good way to put it, yes. Take cancer again. We often think of it as a disease of the genome, mutations, deletions, rearrangements in the DNA, and that's definitely part of the story. But tumorigenesis, the process of a tumor developing, is almost always accompanied by massive changes in the epigenome too. These epigenetic shifts mess with cell identity. They can alter how the immune system sees the tumor, and they cause huge changes in the transcriptome, which genes are actually being turned on or off. What's really striking is that most cancers actually have mutations or alterations in both the genome and the epigenome. They're often intertwined. Studying both is critical.
Okay, this is the perfect setup then. Let's unpack this further. We're diving right into Chapter 7 now, specifically histone modifications. Let's start right at the beginning. What are histones? Why are they and their modifications so central to all this?
Right. So imagine your DNA. It's this incredibly long, thin molecule, right? About 2 meters of it packed into each cell nucleus, which is microscopic, maybe 10 micrometers across. To make that fit, it needs to be packaged extremely efficiently. That's where histones come in. Histones are a family of proteins. They're positively charged, which helps them bind tightly to the negatively charged DNA. The DNA wraps around these histone proteins, forming these little bead-like structures. We call these fundamental units nucleosomes. Think of DNA as the thread and histones as the spools it wraps around.
Okay, the spool analogy works, so it's wound up tight.
It's wound up, yeah. And these nucleosomes, these beads on a string, are the basic building blocks of chromatin. Chromatin is the whole complex DNA plus all the associated proteins, mostly histones, that makes up our chromosomes.
So the packaging isn't just about cramming it in, it's functional.
Absolutely functional. The way the DNA is wrapped around these histone spools is not static. It can be dynamic. If the chromatin is tightly packed, condensed, the cellular machinery that needs to read the DNA can't easily get access. Those genes are effectively silenced. If the chromatin is more open, relaxed, then the machinery can get in and the genes can be expressed. And this is where the modifications become absolutely crucial. Chemical tags can be added to or removed from the histones themselves, particularly their tails that stick out from the spool. These modifications act like signals.
Signals that form a kind of code. You mentioned the histone code.
Exactly. It's not just random decoration.
Yeah.
These modifications form a complex language, often called the histone code. It's like a set of instructions written directly onto the packaging material. These instructions tell the cell machinery, this region should be active, or this region needs to be shut down, or even this region is poised, ready for action later.
A code. That sounds complex. What kinds of modifications are we talking about? What are these chemical tags?
There's a whole variety, but some of the most studied and important ones are post-translational modifications, or PTMs. That just means they happen after the histone protein itself is made. Key examples include acetylation adding an acetyl group and methylation adding a methyl group. Methylation can even be mono, di, or trimethylation, adding 1, 2, or 3 methyl groups, and each might mean something different. There are others too, like phosphorylation, ubiquitination. It's a diverse toolkit of signals.
Okay, so you have these tags, these methyl or acetyl groups. What do they actually do? How does adding a tag change gene activity?
Right, the so what? It's profound. These modifications do 2 main things. First, they can directly affect how tightly the histones pack together and thus how accessible the DNA is. Acetylation, for instance, tends to loosen things up, making DNA more accessible. But perhaps even more importantly, these modifications act as specific binding sites for other proteins, what we call effector proteins. These effectors can be chromatin-modifying enzymes themselves or chromatin remodeling complexes that physically move nucleosomes around. So the histone mark acts like a docking station. An effector protein recognizes the mark, binds to it, and then carries out a specific function, like opening up the chromatin or recruiting the transcription machinery.
So the modification itself doesn't always do the job. It calls in the specialists.
Often, yes, it's a recruitment platform. And here's where it gets really sophisticated. It's often not just one modification in isolation. Multiple histone modifications often act together, in combination, like chords in music rather than single notes. This combinatorial complexity generates very specific chromatin structures. And that specific structure ultimately determines the precise expression level for different sets of genes. It allows for incredibly fine-tuned control.
Wow. Okay. A symphony of signals, like you said.
Yeah.
But if it's that complex across the entire genome,
Yeah.
how do scientists even begin to read this code? How do you map these modifications everywhere?
Yeah, it's a huge challenge. It requires massive efforts, what we sometimes call big biology projects. Thankfully, we've had major international collaborations tackle this. You might have heard of the ENCODE project or the Roadmap Epigenomics project or the International Human Epigenome Consortium, IEEC. These groups have put in tremendous work to generate comprehensive genome-wide maps. They map where specific histone modifications occur, where chromatin is accessible, where transcription factors bind, what RNA is being produced. And they do this across many different human tissues and cell types.
So they're creating these detailed epigenetic atlases for different parts of the body. What have these atlases revealed? Any key patterns?
Absolutely. By integrating all this data, the histone marks, the gene expression, the chromatin structure, we've uncovered really strong correlations and novel relationships. We can now start to associate specific histone marks with particular functions or genomic locations. For example, one mark, H3K4Me3, that's trimethylation on lysine 4 of histone H3, is almost always found right at the start sites, the promoters, of genes that are actively being transcribed. Think of it as a bright green light signaling go for gene expression.
Okay, H3K4Me3 means active promoter. Is there a corresponding stop signal? Yes. Red light?
There is. A very prominent one is H3K27A3 trimethylation on lysine 27 of histone H3. This mark is strongly associated with gene repression, often silencing large regions of the genome, so that's your red light. And there are others. H3K9E3 and H4K20Me3 are often found near the boundaries of tightly packed silent regions called heterochromatin. Then you have marks like H3K9E1 or H4K20Me1, mostly in active gene bodies. Acetylation marks like H3K9AC or H3K27AKE are also generally linked to active regions, particularly enhancers, which are like regulatory switches often located far from the gene itself. Some marks are even linked to DNA replication timing. It's a rich vocabulary.
You mentioned earlier something called bivalent domains, where you have both active and repressive marks together. That sounds contradictory.
It does sound contradictory, but it's a fascinating reality. These bivalent domains have both H3K4Me3, the go signal, and H3K27Me3, the stop signal, coexisting in the same region, often at the promoters of key developmental genes, especially in embryonic stem cells. The idea is that these genes are held in a poised state. They're not fully on, but they're not fully off either. They're ready to quickly go one way or the other when the cell receives the right differentiation signal. It provides developmental flexibility. But this also raises an important question. What happens if that balance is disturbed? Exactly. What happens if the regulation of these crucial bivalent domains goes awry? Well, it can lead to developmental defects or contribute to diseases like cancer, where cells might inappropriately reactivate developmental programs or fail to differentiate properly. And one more function, histone modifications can also act as boundary elements. Like insulators, they can help prevent the spread of silent heterochromatin into active gene regions or vice versa, helping to maintain the proper organization of the genome into functional domains.
Okay, this paints a really dynamic picture. We have these modifications, this language. But who's doing the writing and erasing? You mentioned enzymes.
Right. It's not magic. It's biochemistry. These covalent modifications on histones, and also modifications like methylation directly on DNA, are tightly controlled by specific enzymes. We often group these enzymes into three main categories: writers, erasers, and readers.
Let's start with the writers. Who are they putting the marks down?
The writers are the enzymes that add the epigenetic marks. So for DNA methylation, the writers are the DNA methyltransferases, DNMTs. For histone modifications, the writers include histone acetyltransferases, HATs, which add acetyl groups, and lysine and methyltransferases, KMTs, which add those methyl groups to lysine residues on the histone tails. They literally write the code.
Makes sense. And the erasers.
Yeah.
Taking the marks off.
Exactly. Epigenetic marks need to be dynamic, removable. The erasers are the enzymes that remove these marks. For DNA demethylation, we have enzymes like the TEAT proteins. For histones, we have histone deacetylases, HDACs, which remove acetyl groups, and lysine demethylases, KDMs, which remove methyl groups. This constant process of adding and removing marks allows the cell to respond to changing conditions.
Okay, writers add, erasers remove. What about the readers? What's their role?
The readers are crucial. They are proteins that don't modify the histones themselves, but instead recognize and bind to specific modifications. They possess specialized domains that act like little hands grabbing onto a particular histone mark, like H3K4ME3 or H3K27ME3. Once bound, these reader proteins then recruit other factors or initiate downstream signaling cascades. They essentially interpret the code written by the writers and erased by the erasers. What's really key here is the constant interplay, the dynamic balance between these writers, readers, and erasers. It's this coordinated action that ultimately controls gene expression with such precision.
It's like a whole molecular team managing the genome. Can you give us a bit more on how these readers actually recognize the marks? What are these specialized domains?
Sure. There are several families of these reader domains, but three major types are particularly well studied: chromodomains, PhD fingers, and bromodomains. Each type has evolved to recognize specific types of histone modifications.
Do they differ in what they recognize?
Yep.
Specificity?
They do. Chromodomains and PhD fingers tend to be quite specific, often recognizing particular methylated lysine residues. For example, the polycomb repressive complex contains proteins with chromodomains that bind to H3K27E3, helping to maintain gene silencing. CHD1 is another chromodomain protein. Bromodomains, on the other hand, are generally readers of acetylated lysines. They tend to be a bit less specific about which lysine is acetylated, often recognizing multiple acetylated sites. Proteins like BRD2 or the well-known BRD4 contain bromodomains and are typically associated with active transcription, linking histone acetylation to gene activation. So you have readers for activating marks and readers for silencing marks, allowing the cell to translate the histone code into specific functional outcomes.
Okay, so putting it all together, our cells are constantly getting signals from outside, right? Hormones, growth factors, nutrients. How do these external signals translate into changes in gene expression using this histone modification machinery?
That's exactly where this system shows its power. Those signals from the extracellular matrix, from cytokines, peptide hormones, nutrients, various compounds, they don't just float around. They trigger signaling cascades inside the cell that ultimately converge on the chromatin. These signaling pathways can activate or inhibit the writers and erasers we just talked about. So a signal might lead to a HAT being activated, increasing acetylation at certain genes, or an HDAC being recruited, removing acetylation. The histone tails essentially act as integration platforms, receiving and processing these incoming signals. And what's really cool is that multiple signals occurring over time can leave their mark essentially being stored on the chromatin.
Stored information.
Mm-hmm.
Again, this idea of memory. So the chromatin remembers the signals it received.
In a way, yes, it builds up a history. And this dynamic epigenetic landscape reflecting past and present signals is constantly being interpreted by those reader proteins. The mechanisms are complex. A modification might change how well a reader binds nearby, or it might recruit an enzyme that adds another mark. Or a combination of marks might create a unique surface recognized by a specific complex. The end result of all this signal integration and interpretation is the transcriptional output, which genes get turned up, which get turned down, ultimately changing the cell's state or behavior in response to its environment.
It sounds incredibly precise. How does the cell fine-tune this, maintain that balance between active and inactive genes?
It's a constant balancing act. Take acetylation again. The writers, HATs, and erasers, HDACs, are often both present near active genes. Their levels and activity correlate with how much mRNA is being produced and how much RNA polymerase II, the main transcription enzyme, is there. This ongoing antagonism, this push and pull between HATs and HDACs, allows for very precise fine-tuning of gene expression levels. It maintains the homeostasis of active chromatin regions, both at the start sites, TSS, and at regulatory elements like enhancers. Active genes generally live in this more open state called euchromatin.
What about genes that need to be ready but aren't active right now? You mentioned poised genes earlier.
Right, poised genes. These are fascinating. They aren't actively transcribed, and they lack high levels of the typical activating mark, histone acetylation. But they often do have the activating H3K4 methylation mark, sometimes alongside the repressive H3K27 mark, that's bivalency, and often incorporate a specific histone variant called H2AZ. They tend to reside in a state called facultative heterochromatin, not fully open, not fully closed. The idea is that there's a kind of dynamic equilibrium here too, perhaps a cycle of very transient acetylation and deacetylation. This keeps the gene primed but inactive, holding its promoter region in a ready state, waiting for the right signal to trigger full activation. It's cellular preparedness.
This concept of storing information, the poised state,
Hmm.
it really hammers home this idea of epigenetic memory. Can you expand on that? How stable is this memory?
Yes, epigenetic memory is a really central concept. It refers to the ability of cells to maintain their identity and gene expression patterns over long periods, often through many cell divisions. It's how a liver cell stays a liver cell for years. This long-term stability relies on robust gene regulatory networks and, crucially, the preservation of specific epigenetic states.
And what's the molecular machinery behind that long-term memory? Is it different from the short-term responses?
It seems to be, yes. The long-term, mitotically heritable memory, the kind passed down when cells divide, seems to rely heavily on the actions of chromatin modifiers within specific large protein complexes, particularly the polycomb group, PCG, and trithorax group, TRIRXG complexes. PCG complexes are generally involved in maintaining silent states, often associated with that H3K27Me3 mark. TRIRXG complexes are involved in maintaining active states, often linked to H3K4Me3. Interestingly, the core players in these long-term memory complexes are often the lysine methyltransferases, KMTs, and demethylases, KDMs, the enzymes dealing with methylation. The HITs and HDACs involved in acetylation seem less central to maintaining these very long-term heritable states.
So methylation for long-term memory, acetylation more for short-term responses. Is that a fair simplification?
It's a useful simplification, yes. It reiterates this principle. The rapid day-to-day adjustments of the epigenome in response to immediate signals often involve changes in histone acetylation levels. These are generally quite dynamic and perhaps less stably inherited, whereas the really fundamental long-term decisions like establishing cell fate during differentiation or perhaps storing memories of significant past events like infections, these seem to be encoded more robustly in patterns of histone methylation. These marks provide a more durable form of epigenetic memory. Understanding this difference is key.
It really underscores how critical getting this programming right must be, right from the very start of development.
Absolutely critical. During early embryogenesis, there's this massive wave of epigenetic reprogramming. The epigenome inherited from the sperm and egg gets largely wiped clean and reset. This resetting establishes naive pluripotency, allowing the early embryonic cells maximum developmental potential. And there seems to be a hierarchy here. Often, changes in histone modifications happen before changes in DNA methylation. Histone marks might guide the way, and DNA methylation comes in later to lock things down more permanently.
That fits with the Waddington landscape analogy, doesn't it? The ball rolling down the hill, guided into valleys of cell differentiation by the epigenetic landscape.
Exactly. Waddington's landscape is a fantastic conceptual model. Once a cell differentiates, it's like the ball has rolled into a deep valley representing, say, a neuron or a muscle cell. The walls of that valley are maintained by the specific epigenetic state, the pattern of DNA methylation, histone modifications, and even the 3D folding of the chromatin. This stability prevents the cell from easily rolling back up the hill or jumping into a different valley under normal conditions. It ensures tissue integrity.
But what if those valley walls crumble? What if homeostasis is disturbed by, as you call them, epimutations?
Right, that's the crucial question for disease. What happens when this carefully maintained epigenetic landscape is disrupted? If the chromatin becomes too rigid, too restrictive, like the valley walls are too high, it might prevent necessary cell state changes, like stem cells differentiating when needed. Conversely, and perhaps more relevant for cancer, if the chromatin becomes too permissive, too loose, the valley walls are too low, cells might de-differentiate or switch lineages inappropriately or fail to stop dividing. These deviations from the normal epigenetic state are now recognized as major contributors to tumorigenesis. The balance is vital.
That connection to cancer is clear. But what about other aspects of memory? Does epigenetics explain how our bodies seem to remember lifestyle factors or past infections, maybe even influencing aging or future generations?
Yes, the scope is incredibly broad. The epigenome acts as a memory keeper in many contexts. Our neurons obviously store cognitive memory, likely involving epigenetic mechanisms for long-term storage. Our immune system exhibits memory too after an infection. Certain immune cells remain altered, poised to respond faster and stronger next time. This is called trained immunity, and it has an epigenetic basis. And our metabolic organs, muscle, fat, liver, they seem to epigenetically remember long-term patterns of diet and exercise influencing our metabolic health.
And how does this relate to aging? You mentioned epigenetic drifts.
Right. Over our lifespan, our epigenomes don't stay perfectly static. They undergo subtle cumulative changes, often called epigenetic drift. This drift can be influenced by our experiences, our environment, our lifestyle. These changes accumulate in our somatic cells, or body cells, and are passed down when those cells divide. They form a kind of lifelong record, and critically, epigenetic alterations are now considered one of the fundamental hallmarks of aging. Things like global changes in DNA methylation, alterations in histone modification patterns, loss of histones, changes in chromatin structure, these all contribute to the aging process and age-related decline. And these changes are often heavily influenced by environmental factors and nutrient availability, linking lifestyle directly to the pace of epigenetic aging.
That's somatic cells. What about the next generation? You mentioned transgenerational inheritance.
That's the really provocative area. The idea is that some epigenetic marks acquired in the germ cells, the sperm and egg, might potentially escape that reprogramming phase after fertilization and be passed down to the offspring. This raises the possibility of transgenerational epigenetic inheritance, where, for example, a parent's or even grandparent's diet or exposure to stress could influence the health or disease risk of descendants via epigenetic mechanisms. It's a complex and sometimes controversial field, but the evidence is growing and the implications are profound.
Okay, let's circle back specifically to cancer and histone modifications.
Hmm.
We know epigenetic changes happen, but how exactly do histone modifications go wrong in cancer, and what are the therapeutic angles?
Right, it's a major focus. Altered histone modification patterns are key features of many cancers. We see broad changes, like often a global decrease in certain repressive marks, which might destabilize the genome.
Things get looser where they shouldn't.
Exactly. But we also see very specific changes. For example, the promoters of tumor suppressor genes, genes that normally put the brakes on cell growth, often gain repressive histone marks like H3K27ME3 or H3K9IME3, effectively silencing these crucial protective genes. Sometimes, this epigenetic silencing of a DNA repair gene can be an early event, making the cell more prone to accumulating actual DNA mutations later on. One epimutation can unleash genomic chaos. And it's not just the marks themselves. The enzymes, the writers, erasers, and readers are frequently mutated or misregulated in cancer. Genes encoding KMTs, writers of methylation, KDMs, erasers of methylation, HATs, writers of acetylation, and HDACs, erasers of acetylation, are among the most frequently mutated genes across various cancer types. For instance, mutations might cause a KMT like EZH2, which writes the repressive H3K27A3 mark, to become overactive, leading to excessive gene silencing. Or mutations might inactivate a KDM that normally removes repressive marks.
So the machinery itself is broken, leading to a faulty code.
Precisely. Or reader proteins can be affected. You might get overexpression of a reader like BRD4, which binds acetylated histones associated with active genes, potentially driving uncontrolled expression of cancer-promoting genes. And sometimes, even the histone proteins themselves are mutated. There are now famous examples like specific mutations in histone H3 variants like H3.3 found in certain pediatric brain tumors and bone cancers. These oncohistone mutations directly interfere with the writing of other nearby modifications, disrupting normal gene regulation.
That's a complex mess of potential problems. But you said epigenetic marks are reversible. Does that offer hope for treatment?
That's the crucial point, yes. Because these modifications are enzymatic and reversible, unlike permanent DNA mutations, they represent very attractive therapeutic targets. This has spurred the development of epidrugs. These are typically small molecule inhibitors designed to target the activity of specific epigenetic enzymes, the writers, erasers, or even the readers. Oncology is really leading the charge here. We now have drugs targeting HDACs, DNMTs, and increasingly, the more specific KMTs and KDMs.
Can you give a specific example of an epidrug targeting histone modifications that's in use?
A good example is tazemetostat. It's an inhibitor of EZH2, that KMT we mentioned, that writes the repressive H3K27Me3 mark. EZH2 is often overactive in certain lymphomas and sarcomas. By inhibiting EZH2, tazemetostat reduces the levels of H3K27Me3, which allows previously silenced tumor suppressor genes to become re-expressed, helping to slow down or stop cancer cell growth. It's approved for treating specific types of sarcoma and lymphoma. And the hope is that targeting methylation modifiers like KMTs and KDMs might offer more specificity and potentially fewer side effects than the older, broader inhibitors of acetylation or DNA methylation, because methylation marks tend to have more specific roles.
It's not just cancer, though. Are histone modifications implicated in other types of diseases like neurological disorders?
Yes, definitely. Histone acetylation in particular has been strongly linked to several neurological and neurodegenerative conditions. There seems to be a disruption in the balance between HATs, adding acetylation, and HDACs, removing it, in diseases like Huntington's, Alzheimer's, and others, leading to abnormal gene expression in neurons.
Is there a clear example where this is the primary cause?
A classic example is Rubinstein-Tybee syndrome. It's a rare genetic disorder characterized by developmental delays and intellectual disability. It's caused by mutations in genes encoding AKTs, specifically CREBP or EP300. Because these patients can't properly acetylate histones, they have widespread gene dysregulation. And this immediately raises a therapeutic question.
Can you compensate? Maybe by blocking the removal of acetylation?
Exactly. If the problem is too little acetylation because the writer is broken, maybe you can restore levels by inhibiting the eraser. HDAC inhibitors are being explored as a potential therapy for Rubinstein-Tybee syndrome. Similar ideas are being tested for other conditions where gene silencing is a problem, like Friedreich's ataxia or Fragile X syndrome. Using HDAC inhibitors to try and reawaken silenced genes is a really active area of research.
Oh, fascinating. Okay, one more connection I want to make. Diet. We eat every day. Can the food we eat actually influence our epigenome, these histone modifications? Is there a direct link?
Yes, absolutely direct. This is the field of nutritional epigenetics, a part of the broader field of nutrigenomics. The link is often through metabolism. Many of the enzymes that write and erase epigenetic marks, HATs, HDACs, KMTs, KDMs, TTs, require essential metabolites as co-substrates or cofactors. Think about acetyl-CoA, which is central to energy metabolism and is the source of the acetyl group for HATs, or S-adenosylmethionine, SAM, the universal methyl donor for KMTs and DNMTs, derived from folate and B vitamins, or alpha-ketoglutarate, NAD plus ATP, all key metabolites that directly fuel epigenetic modifications. So your metabolic state, which is heavily influenced by diet, directly impacts the availability of the building blocks and fuel needed for epigenetic marking.
So nutrients literally provide the raw materials for the epigenetic machinery.
Yeah.
What about specific compounds in food?
Beyond the core metabolites, yes. A huge range of bioactive compounds found in plant-based foods, fruits, vegetables, teas, spices can also directly influence epigenetic processes. We're talking about phytochemicals like genistein from soy, resveratrol from grapes, curcumin from turmeric, EGCG from green tea, sulforaphane from broccoli. The list is long. These compounds can directly inhibit or activate epigenetic enzymes like DNMTs, HATs, or HDACs, or influence signaling pathways that control these enzymes. So diet provides both the fundamental building blocks and specific modulatory signals for the epigenome. And with modern omics technologies, we can now study the effects of these dietary compounds on a genome-wide scale, assessing their potential for disease prevention or even treatment, particularly in areas like cancer. It really highlights how diet is not just calories, but information that speaks directly to our genes.
Okay, let's tie all of this together now. We have genomics, the DNA sequence, we have epigenomics, these histone modifications, and other layers of regulation, and we have these powerful sequencing technologies. It really feels like this is converging towards truly personalized medicine.
That's exactly where it's heading. The explosion of next-generation sequencing, NGS, has been revolutionary. It allows us not just to sequence a person's genome relatively cheaply, but also to profile their epigenome, transcriptome, proteome, metabolome. This leads to the idea of integrative personal omics profiling, or IPOP. It's about combining your static genomic blueprint with dynamic longitudinal monitoring of all these other molecular layers that reflect your actual physiological state in real time.
Real-time monitoring of your ohms. What can that actually tell you? Any concrete examples?
There have been proof-of-principle studies that are quite striking. What's fascinating is that by taking frequent samples, blood draws, etc., over time, these IPOP studies could detect subtle, personalized shifts in physiology long before symptoms appeared. In one well-known study, they picked up markedly elevated glucose levels in one individual specifically following viral infections. This very early detection of a drift towards a pre-diabetic state allowed that person to intervene effectively with diet and exercise changes and actually reverse it. Imagine scaling that kind of early detection. And as we've discussed, oncology is already deeply involved in this. Understanding the specific driver mutations, including epigenetic alterations in an individual's tumor, is becoming standard practice for guiding treatment. The old one-size-fits-all chemotherapy approach is increasingly being supplemented or replaced by targeted therapies based on the molecular profile of the tumor. Precision oncology relies heavily on reading both the genome and the epigenome. And this personalization should make developing new drugs more efficient, too. Absolutely. If you can stratify patients based on their specific genomic or epigenomic profile, clinical trials can be designed more intelligently. You might need fewer participants because you expect a larger effect size from a treatment tailored to the right subgroup. And this applies beyond cancer. Think about complex diseases like type 2 diabetes. We're moving towards subtyping T2D based not just on symptoms, but on underlying genetic and epigenetic factors, and how individuals respond dynamically to different diets or interventions. This allows for truly personalized prevention and therapy strategies. So you can really think of epigenomics as the vital second dimension of genomics. Genomics gives you the parts list, the DNA sequence. Epigenomics tells you how those parts are being used, regulated, and organized in real time and over your lifetime. These patterns, DNA methylation, histone modifications, they organize the 3D structure of the genome in the nucleus, they control which genes transcription factors can access, and they maintain that crucial epigenetic memory. And remember, the long-reach things like nutritional exposures in utero during fetal development can leave lasting epigenetic marks that influence risks for obesity, diabetes, metabolic syndrome decades later. It shows how powerfully the epigenome connects our past, present, and future health.
It's truly remarkable. We've covered so much ground today, really diving deep into histone modifications. We've seen how these, well, seemingly tiny chemical adjustments to DNA's packaging proteins are actually fundamental regulators. They control cell identity, store forms of memory, and critically influence our health and disease risk.
It really is an incredibly intricate and dynamic system, isn't it? It goes so far beyond the static DNA sequence. It's this constant dance between the writers, readers, and erasers of the epigenetic code, constantly interpreting signals from inside and outside the cell, integrating our history, shaping our present function. It's really the key to cellular adaptability and identity.
In understanding this layer within our broader genomic medicine program, it really feels like it's unlocking a new era in health. The ability to potentially read, interpret, and maybe even influence our own epigenomes. It opens up truly exciting possibilities for preventing and treating diseases in ways that are much more personalized and precise.
That's the ultimate goal, isn't it? Connecting all these dots, from the genome to the epigenome to lifestyle and environment. It points towards a future where healthcare isn't just reactive, but proactive and deeply personalized, where interventions, whether it's diet advice or drug therapy, are tailored specifically to your unique biological makeup, considering both your genes and your epigenetic patterns.
So as we wrap up, here's something for you, our listener, to think about. Considering the profound role your epigenome, especially these histone modifications, plays in remembering your experiences and shaping your future health, what lifestyle choices could you make starting today that might help write a healthier epigenetic story for your own tomorrow?
And perhaps on a broader note, this raises an important question for all of us. As we gain deeper insights into our personal epigenomes and how our choices affect them, how might our understanding of individual responsibility and our approach to proactive health management continue to shift and evolve in the years to come?